All right, well, welcome to our last installment in this particular chapter, looking at this point, mutations or changes in DNA that can occur and what it will do to the resulting proteins. Now, we know a mutation is simply a change in the bases of a DNA molecule. Now, earlier in the semester, we looked at chromosomal mutations that can occur, where whole segments or large portions of a chromosome are deleted or inserted or reversed or whatever the case may be. At this point, we're going to focus on single genes and specifically point mutations, where a single base pair can be altered, and that can ultimately lead to new functions of proteins or non-functioning proteins. Now, there are two basic types of point mutations where you substitute or alter a single base pair. There are substitutions where one base replaces another, and then there are insertions or deletions where one base is deleted or added to the DNA molecule. So again, to sort of summarize, a point mutation is a change in a single base pair, whether you add, delete, or substitute. Now, this is a nice way to look at some of these different types of mutations that can occur. Here, the original DNA sequence being read from 5' to 3' is TTC. Okay, here is a specific substitution where you alter one of the bases, alter an A and T, they get switched around here. And as a result of that, the messenger RNA that's synthesized has been altered, and ultimately the amino acid that is produced will be altered as well. And in sickle cell anemia, a single base pair alteration can lead to drastic changes in the shape of the hemoglobin molecule. Now, are all mutations detrimental? Well, not quite. Sometimes you can have a mutation that occurs, as in this case here, an A replaces the G from the original strand. Well, because of the redundancy in the genetic code, the amino acid that gets produced is still the same as the amino acid on the wild type or non-mutated DNA strand. So there are instances where the bases that get changed around still lead to the addition of the same amino acid in the same location. So it has no effect on the amino acid sequence of the resulting protein. So again, it's deemed a silent mutation. Now, occasionally bases are altered, and that leads to the addition of a different amino acid. In the original strand of the wild type, you get a glycine. Here you get a serine. Now, this is termed a missense mutation. You're making something, but you're making the wrong strand. You've altered the strand in some way, shape, or form. Now, is this bad? Well, again, the answer is it depends. If this occurs on a non-coding part of the DNA molecule, nothing happens. Now, if a mutation like we see here occurs on the coding portion of the DNA molecule, well then, yeah, potentially you're going to change the order of the amino acids, but again, it sort of matters. The active site is what's obviously actively involved in the chemical reactions that are catalyzed. So if a mutation is away from the active site and doesn't have significant impact on the function of the protein, you can have a missense mutation that is still minimally effective or minimally alters phenotype. Now, there are worse case scenarios, also worst, I guess, but we're talking about something that's worse, okay? Here we have the wild type. There's your amino acid sequence or your polypeptide that's being created. Occasionally, a mutation can occur that leads to a premature stop codon. Now here we have a polypeptide that is 4 amino acids long, but because this particular mutation causes a stop codon to occur earlier, you have a truncated or shortened polypeptide, and that is almost always detrimental, always leads to non-functioning proteins, and is definitely bad news. So nonsense mutations lead to shortened proteins because you get an early stop codon. Now, frame shifts are when you insert or delete bases. So here we have TAC-T. Well, what's happened is we have TAC, there's T, but we've added in an adenine here. The result of that is, again, you change the reading frame. So we have this initial reading frame that's AUG, AAG. It's now AUG, UAA, and then there's a G. So there is, here are the codons in the original reading frame. They've all been shifted over because of the insertion of this base. So this is a particular frame shift mutation. When you insert or delete a base, it changes the reading frame, and this causes a nonsense mutation. Let's see, here we've removed a base. So you remove a letter, and again it changes the order of the amino acids, changes the reading frame because the reading frame has changed, and again you're still making a polypeptide, but it's the wrong polypeptide, so it's called a missense. So just remember, nonsense ends the polypeptide early, missense, you make the polypeptide, but you have a lot of the wrong bases. In either case, there's a high probability of non-functioning proteins. Let's see. Now, here's another interesting sequence. What if you removed 3 bases? What if you remove one of the codons from the reading frame? Well, here methionine, lysine, phenylalanine, glycine. Because we remove one codon, we still get many of the same amino acids. You just have lost one particular amino acid from the polypeptide. Now, this can certainly be detrimental, but it tends to have less of an effect on the resulting protein than a frame shift mutation where you add or delete 1 or 2 bases. So it seems sort of odd, but missing 3 bases in a row or missing a codon oftentimes has less of a negative impact than adding or deleting 1 or 2 bases. So again, substitutions occur when you replace one nucleotide or one pair of nucleotides with another. Silent mutations, there's a mutation, but you still get the same amino acid, so you still get the same resulting protein. We don't notice any problems. Missense mutation, we still get a polypeptide, but it's not necessarily the right sequence of amino acids. And then a nonsense mutation, again, you get an early stop codon, and that's big problems. Let's see, insertions or deletions, again, we're adding or losing nucleotides, oftentimes with deleterious effects, so it's harmful. And this is what ultimately we call frame shift. Okay. Now, what can cause these issues? The class of factors that can lead to mutations are called mutagens. Mutagens, it could be physical, like ultraviolet radiation. So, radiation is a physical mutagen. It can damage the bases. Or you can have chemical agents like what you see in cigarette smoke cause mutations. So you can have physical or chemical factors that can lead to mutations, and again we call them mutagens. Just nice image of the process here. Okay, so here's the big idea. We all get down to this. What is a gene? Okay, well, we want to sort of expand our definition of a gene. So a gene, it's part of a DNA molecule, so that's good. But we say it produces a final functional product. We don't say one gene produces one enzyme, or one gene produces one protein, or one gene produces one trait. Think of it more broadly in terms of some product that is produced and can be used. Now, it could be a polypeptide that will make a protein, be it a structural protein or an enzymatic protein, or you could even come up with RNA molecules like ribosomal RNA. Let's see, tRNA or transfer RNA, or those ribozymes that we talked about. So genes are segments of DNA that make something functional, whether it's something related to a protein or RNA. So to think of our broadest definition of a gene, our most inclusive definition of a gene, it's a discrete unit of inheritance. That was Mendel's idea here. It's something that gets passed on from parent to offspring and influences traits. We can look at a gene as being a nucleotide sequence or sequence of DNA on a chromosome. That's what Hunt Morgan looked at with his fruit fly experiments. Or we can look at DNA as a sequence that codes for some particular polypeptide, and that is more of our molecular DNA perspective that we looked at in this particular chapter. Now, this chapter takes some work, it takes some study, but think big, use the animations, and try and look at how all these pieces work together to produce polypeptides. Remember, it's DNA to RNA to protein. Keep the big idea in mind, get help when you need it, and you'll certainly do fine. All right, well, good luck.